Thank you for the introduction. Dear members of the Scientific Council, dear committee members, dear reviewers, thank you for giving me the opportunity to give this presentation here on the grounds of the Czech Technical University. I'm really thrilled I could speak here about my research. Also, thanks to all those watching on YouTube for watching on YouTube. So, as you could see from my title, my research topic is somewhere between the very technical one and pretty medical one. So I know that most of you here have a technical education, but to give you a really good overall picture of my research, I'll focus more on the clinical applications rather than on the technical details. So I promise to keep the medical details low so that hopefully it's nicely understandable for everybody. So the talk and my research is mostly about arterial spin labeling, which is an imaging modality for imaging perfusion. So let me first explain a bit what I'll be talking about. So first, I'll explain some basics of what perfusion is, what is it useful for, how we can measure that, what are the challenges associated to working and analyzing this data, what are the applications, and I'll also later on talk about my current research and my future work. So, perfusion. For simplicity, I will focus only on the perfusion in the brain, as this is the main topic of my research. So, as everybody knows, brain is in constant need of uninterrupted supply of oxygen, sugar, and other nutrients. So all these are delivered to the brain through blood by a network of vessels. So there are several main arteries coming through the neck, supplying blood to the brain. These arteries further branches to smaller vessels and then to capillaries. It's in these capillaries that the actual exchange of the nutrients and oxygen between the blood and tissue happens. And it's the supply of blood, so the amount of blood coming to this capillary bed expressed in milliliters per minute per 100 gram, this is what we call perfusion. Now, I said that perfusion, we need an uninterrupted supply of blood to the brain, but that doesn't mean that the perfusion has to be constant at all times. Actually, there's quite some variation between individuals and also within an individual, and also perfusion changes in time. So this gives us a very nice parameter to study the healthy population. As an example, we can study the brain activation in some regions related to a task, because when the brain is active, the perfusion goes up in the related region. Also, perfusion changes with age. For older subjects, it decreases compared to the younger population, and this gives us a good parameter to study normal aging. Also, perfusion is a good parameter to study any issues with the development of the brain or some chronic problems. So, for example, we have compared a population of young kids that were suffering from malnutrition during gestation, and even 60 years after that, we can see differences in perfusion between the group that was affected or wasn't affected. So perfusion gives us a very strong parameter to study many different aspects of the healthy brain. Of course, it is also a very useful parameter for studying diseases of the brain. So, for example, perfusion decreases in neurodegenerative diseases. There are very specific patterns of perfusion decrease in Alzheimer's disease. Very obvious focal regions of hypoperfusion are typical for stroke, as you can see here, but also we can see later blood arrival to regions that are supplied by collateral vessels, which are alternative routes that sort of start supplying the brain in case that the main supply is blocked. An example of an increased perfusion is a patient with a brain tumor here. So in certain types of brain tumors, we can see increased perfusion in the active part of the tumor, which is related to having many new vessels formed in the tumor, and it's a hallmark of tumor aggressivity. So my main goal, the goal of my research, is to study perfusion on all these diseases and also in the healthy population, and try to turn perfusion into a clinical biomarker, which can then later be used for early screening of diseases, disease localization, assessing the severity of a disease, or for therapy monitoring. So now, of course, any useful parameter is not useful enough if there's no simple and easy way to measure it. So there are several modalities that allow measuring perfusion. For example, we can inject radioactive water to vessels and then measure it using perfusion emission tomography. We can inject a high-density tracer and measure it with computed tomography, or we can inject gadolinium-based tracer and measure with MRI. Now, you should be asking, like, there's not arterial spin labeling mentioned here, and so why we should develop a new method when there are already 3 very well working methods. And the issue is that, of course, all methods have some drawbacks. So of all of these methods, they rely on injecting exogenous tracer to the body. PET and CT, they expose the patient to ionizing radiation. It's nearly impossible to do water PET in hospital in real patients. And while DSC MRI is used very often, it brings certain issues, like there are concerns about gadolinium deposition in the body, and it also brings some extra costs. Moreover, DSC cannot allow absolute quantification of the flow, and is not easily repeatable within the same session. So this is where arterial spin labeling comes in, as it offers solution to most of these issues. It's completely non-invasive. It doesn't require injection of any ionizing or non-ionizing tracer. It's widely available, easy to obtain, measure, and repeat, and it provides absolute quantification. So to explain how ASL work, I will very quickly say some basic details about nuclear magnetic resonance that is used for measuring ASL. So in nuclear magnetic resonance, we use the property of the nucleon called the spin property. In MRI, we focus almost exclusively on hydrogen protons. So if we place these hydrogen protons to a strong magnetic field, they align with this field, and now they are able to accept energy in form of a photon that is defined by the strength of the magnetic field. After some time, this photon is again re-emitted at this specific resonance frequency, and we can measure the photon, and based on its energy, we can derive some information about the tissue we're measuring. The only disadvantage is that we're just measuring the signal, but we cannot really know where the signal is coming from. So a device that allows us to obtain real images of tissue using nuclear magnetic resonance is called magnetic resonance imaging. You see an example of an MRI machine here. So basically, this is just a very strong magnet. The strength of the field is around 1 or 3 Teslas, which is super high. The main reason to have such a high field is to have enough SNR in the measurement. And as I was saying, any signal coming from the machine is spatially non-specific. So we use a system of gradient coils that can change in time and in space the magnetic field slightly to alter the resonance frequency of the photons. And with that, we are able to encode the spatial position of the signal, and we are able to obtain high-resolution images of the tissue based on their hydrogen content. And of course, MRI is a very versatile modality, so we can alter the measurement sequence to obtain completely differently looking images. So here is an example of an image that measures that is mostly sensitive to content of fat, and this one is mostly sensitive to the water content. Now, with this sequence, we can do even more advanced stuff, and one of them is arterial spin labeling that allows us to measure perfusion. So in arterial spin labeling, we are not injecting anything to the body. Instead, we are using the blood labeled magnetically in the neck as a tracer. So the blood is labeled in the neck, then we wait for 1 or 2 seconds until the blood gets to the brain, and then we take a normal image of the brain. We repeat this measurement 2 times, with and without any prior labeling, and now these 2 images, they look alike, but there's like 2-3% difference between them, and this is related to all the labeled blood that is in one of these images. So if we subtract these 2 images, we obtain an image that is perfusion weighted, and we can quantify this and obtain images of perfusion. Now, unfortunately, there are some challenges related to working with these images. The main challenge is that MRI is traditionally a qualitative method meant to obtain images that are then visually evaluated by medical doctors. Now, with these advanced sequences like arterial spin labeling, ASL, we can do quantitative measurements. And for quantitative measurements, we of course need to know the exact parameters of the measurement. We need to properly quantify it and process the data to have a really good quantitative measurements and not just nicely looking images. So there are 4 basic issues I want to discuss here related to working with ASL data. So the first of them is data standardization. So ASL is mostly a research sequence, and so that means that there are different labs having their own implementation. All the different MRI producers have their own implementation, and while these methods are fairly standardized, they are not really standardized in the way how they save their data and how they save all the parameters necessary for quantification. So it is really difficult to share data if you don't have all the information about the data, because you cannot later quantify it. And of course, this looks like a very simple task. You just take all the measurement parameters and this is what you need. But of course, in practice, this doesn't really work. So people love to publicly share data that don't have any parameters or annotation, or they love to change the protocol in the middle of the study, and so on. So typically, cleaning the data and curating them and getting some use of them is a large part of the analysis. So for that, we decided that we need one clear standard that would allow us to save the ASL data so that we can easily share that. So in collaboration with the Brain Imaging Data Structure Initiative, we've introduced a simple format, simple standard that allows us to organize the data from many patients, different sessions, in a unified way so that it's clear, single way how to save the data. And also with that, we save all the necessary imaging parameters so that, first of all, we know that we have all the necessary parameters and that they are saved in a standardized fashion and allow easy exchange between the sites. A second issue with processing is that, again, it's not completely straightforward to process the data, as especially medical doctors are not really used to work with quantitative data. So we made an EU-wide survey where we were asking medical doctors from hospitals all around Europe what is really, if they are using ASL, and if not, then why not? And most of them said that the lack of tools and training is the main hurdle that stands in their way. So for that reason, I've developed, in collaboration with Amsterdam University Medical Center, an automatic processing pipeline for ASL data called Explore ASL. So this pipeline contains all the necessary steps for processing, from data import, segmentation, co-registration with other modalities, quantification, and analysis. This pipeline is now publicly available on GitHub. Anybody can freely use it for non-commercial research, and we've processed over 20,000 scans with our collaborators using this pipeline. Now, as I was saying, a second issue is also the lack of training. For that, the Open Science Initiative for Perfusion Imaging was created to help users, to help educate users using sequences for perfusion imaging, and also to give them more wider access for tools for processing them. So I lead one of the task forces for ASL within this initiative, and we do many things like comparing pipelines together, making challenges, explaining to people how things work, and we also focus on educational content to provide users with manuals and with training. Now, the last thing I want to mention regarding the technical part is validation and reproducibility. So, of course, all the methods need to be reliable, all the measurement methods. So that means it should be repeatable on several measurements, and we also need to compare it with some standard methods to make sure that we are accurately measuring what we want to measure. So a lot has been done in this field by others, and we have our significant contribution to validation the methods. So for example, we were able to show that ASL perfusion measurement is reproducible even if you use different magnetic field strengths and different resolution. So while these images, they look somehow different, if you properly compensate for all the input parameters and take the resolution into account, they can output similar quantitative numbers. We were also able to show that measuring brain activation with ASL is comparable to the established standard of both functional MRI, and the advantage is, of course, that the functional ASL measurement is able to provide a real, quantitative assessment of how the perfusion is increased following activation. And as a last thing, all those validation studies, they don't only need to be run in healthy subjects and in some easy simulations, but they need to be tested also in the patients. And the reason is that there are certain assumptions under which this quantification of ASL images is done, and these assumptions might be broken in certain diseases. So a separate validation for each different disease has to be done. So we've compared ASL measurement of perfusion in brain tumors to the gold standard PET imaging, and we found that despite slightly overestimating, ASL is sensitive to measuring perfusion also in the brain, while the established technique using gadolinium-based contrast sometimes fails in certain tumors, probably because the vessel density or vessel diameter is not exactly as it should be. So now, if we take all what I've just said together, then we can state we have a pretty good sequence to measure perfusion with ASL MRI. There's also plenty of tools available, so anybody interested can quickly get up to the speed and start using these sequences. So what is really missing now is clinical validation. That means we need to test that the method really works in patients and that it actually brings some benefit that can be used for the good of the patient, for the diagnosis. So we're also working on several validation studies, and I would like to give you 3 examples of how can ASL be beneficial. So this is about how to monitor therapy with ASL, how to use it to discover new information about the disease, and also how to use it as an early biomarker of a disease. So, hopefully in the future we'll be able to work with ASL and develop it as a clinical biomarker, as there are many reasons why we should do that. So first of all, it can replace measurements using tracers or gadolinium contrasts. Also, ASL is really useful as any physiological and metabolic changes in disease might precede structural changes that are typically examined, and also perfusion might be sensitive to different mechanisms in the disease. So as a first example of therapy monitoring, I would like to show you images of with brain tumors. So here we see a resected, operated brain tumor, and the patient with the brain tumor is now undergoing radiochemotherapy of the brain. This is a very aggressive therapy that not only damages the tumor, but also have adverse effects in the healthy tissue, and can even lead to cognitive decline, which is then commonly called the chemofog. So we've looked at these patients to observe what damage is really radiochemotherapy doing in the healthy tissue, not in the tumor, but on the other side. So we've checked how the volume of the tumor changes and how the perfusion of the tumor changes in two groups, one receiving a photon therapy, which has a lot of radiation deposition also in the normal tissue, and with proton therapy that is more focused on the tumor only. What we've discovered is that perfusion decreases that the volume of the tissue decreases by 3% following the radiotherapy. This is dependent on the radiation dose locally, and this then progresses in time. While perfusion decreases abruptly, it's not dependent on the radiation dose, and it doesn't change in time. So we have now two mechanisms of damage to the brain. One, change of a volume that takes some time to build up and then continues also after the treatment, and another one that is visible very early and doesn't change in time. So now if you want to use one of these parameters to quickly judge if the treatment is maybe not so good for the patient, then you probably go for the one that shows very early and doesn't get worse, rather than for the one that takes months to show up in the images. So that is one application of perfusion measurement. In a second application, I want to show two examples from a pediatric population. So first of all is sickle cell disease. So in sickle cell disease, the patients have deformed red blood cells that causes that the red blood cells have a lower oxygen carrying capacity. So we were checking what happens when we measure these patients with ASL perfusion, and we found out that the perfusion is higher in the patients because the body is trying to compensate for the lower oxygen carrying capacity of the blood cells. But interestingly, despite higher perfusion, the oxygen metabolism, so the delivery of oxygen to the brain, is lower. So then we've tried out what happens if we inject a special drug that enlarges the vessels to bring in more blood. So expectedly, the perfusion increases in the patients, but the oxygen metabolism unfortunately doesn't really improve. So that means that no matter how the body is trying to compensate, it cannot really deliver enough oxygen to the brain, which explains why these patients might have problems with not enough oxygen delivery and mini strokes. A second example is craniosynostosis. So this is a genetic disease that leads to children having skull deformations, like pointy heads. And traditionally it was thought that because the skull is deformed, it creates a pressure in part of the brain. This makes it harder for the brain to perfuse it, and thus leads to cognitive deficits, as children very often develop cognitive deficits in these. And so for that, you absolutely have to operate the child. But with perfusion measurements, we managed to show that actually there are no perfusion differences between these kids and normal subjects. So the perfusion, the cognitive deficits that tend to develop in these patients, they are probably not related to mechanistical reasons, but rather to genetic reasons. So this is a part of information that might help to decide what to really do with these subjects, if it's really necessary to operate them or not. And as a last application is dementia. So perfusion is known to be lower in dementia, but of course, knowing lower perfusion in dementia is not very useful, because when somebody has a dementia, we can see that. We don't need to measure perfusion. So we are trying to work developing perfusion as a clinical biomarker of early dementia, so before the subject really starts to show any symptoms. And we did several branches of research on that. We were able to show that the perfusion patterns in the brain are related to the genetics, so more similar between twins than in random people. We were also able to show that perfusion is related to memory decline. Lower perfusion leads to memory decline, also later to vascular damage, and so that early perfusion might really cause later developing dementia. Of course, we are not really too far with that, so the only thing we were able to show that in subjects with genetic predisposition to develop frontotemporal dementia, the perfusion was lower than in their healthy relatives already 10 years before developing symptoms. But unfortunately, none of this really works on individual level, so it cannot be really used for something useful in practice. This is just still research going on, and more data is needed to be able to provide any useful information in the future. So let me just now quickly go through two of the applications we're working on right now. And one of them is mapping of blood-brain barrier. So blood-brain barrier is a structure lining the vessels that effectively regulates the exchange of oxygen and molecules between the blood and the brain. And this effectively helps us to stop toxins from getting from the blood to the brain. Normally, this should work pretty good, but in patients like patients with dementia or brain tumors, this barrier becomes leaky and it starts letting through everything. And our normal ASL sequence, of course, cannot measure this, but if we modify the sequence, we're able to judge if the blood is still in the vessels or if it comes to the tissue, and by that we can calculate proxy parameter that also quantifies the transport across this barrier. So with that, we formed a consortium of over 10 institutes. We plan to measure this blood-brain barrier permeability in healthy patients, in patients with dementia, and with brain tumors. And we have already some promising results. We were one of the first to show that with imaging, that blood-brain barrier permeability increases with age. And as a last project to mention, I want to say something about brain age prediction. So this is a method not developed by us. So the main principle here is that we look at the regional brain volumes, then we train a machine learning method with brain volumes, local brain volumes as the inputs, and the age of a person as the target variable. And with that, we're able to train a model that then, if you put in a subject from the validation data set, it's able to predict its age with an error of about 5 years on average. And of course, it's not really interesting to predict age in healthy people, but people were able to show that this predicted biological age and chronological age differs in disease. So increased aging, as assessed with this method, is a sign of a disease or disease severity in several diseases. And what we are now trying to do is we are combining not only the volumes of the brain, but also the perfusion. So we can take all of these together, all this information, feed them in a machine learning model, predict the age, and see how this improves the model. So we were able to show in healthy subjects that the prediction accuracy goes about half a year better than if you're using just volumes alone. But of course, this is not really interesting. Interesting is to use it in disease and show that there's some benefit in another modality in disease. And so right now we're gathering more subjects. We have around 1,000 or 2,000 subjects for more training. Hopefully we'll get more to be able to use some deep learning on the images itself, and hopefully also apply this to disease. So with that, I would like to conclude my talk. So in general, I hope you get the message that arterial spin labeling perfusion measurement is a repeatable, economical, and safe way to measure brain perfusion. The acquisition and processing is already ready to be used for clinical research, but more validation is needed to really develop some applications that can enter the hospitals. And I would also like to summarize my main contributions to the field. So first of all, we've developed software, helped to validate the method. We had a range of clinical applications to show where this method can be applied. And right now we're working on modeling normal perfusion in large data sets. Thank you.
Thank you very much for a very interesting lecture. Now I open the lecture for discussion and ask you for passing questions. Professor Rybka.
Thank you for the impressive talk. My question is about this polarization pulse used for your method. You told us that the polarization is made in the region of neck. My question is what amplitude of magnetic field you are using for that and what is the duration? And how you arrange the coils so that the field is restricted to the neck region?
Okay, so there's many questions at once. So traditionally, people just label the whole neck with a single pulse. Right now, they're labeling just a very thin plane. You don't really need to adapt the coils, as usually the coil is covering the entire head and also larger part of the neck. So with the normal clinical systems, you can just use a normal coil and it works pretty well. And
what is the duration and amplitude of the pulse, and how you make the polarization field restricted to the neck region?
So restriction of the field is pretty simple. So as I was saying, we have certain magnetic gradients, and we can tune these gradients to change the magnetic field in some space. So let's say we just tune it so that it's increasing from left to right. And now, as I was saying, the tissue, the hydrogens are able to accept energy of photons, but only at certain energy, only at certain frequency. So if we know that we modify the field, then let's say from here to here, we have a certain frequency, and we want to use a radio frequency pulse that matches this frequency. So, well, you cannot really make a nice box radio frequency pulse, but you can get pretty close to that. So with tuning nicely the magnetic field, setting the resonance frequency, and using a correct pulse, you're only able to excite hydrogens in a very thin plane.
What is the amplitude of the field?
I think that's it's like microteslas. It's the gradient field. Amplitude of the radio frequency pulse, not really sure. But you can heat up the subject a bit. So it's like around...
So the polarization field for protons is microtesla?
Sorry, microtesla is... So you use Tesla as the main magnet. This defines your resonance frequency. And then you superimpose extra field with microtesla magnitude to modify it and slightly alter it. Yeah.
Pan professor Tintiera.
No, I just wanted to a bit explain how this works. This is really in microtesla because we use the magnetic part of radio frequency pulse, not the electric part, but magnetic part. So you can express it in microtesla, but usually we call the RF pulse, so we specify the RF pulses by amplitude in let's say volts, and this is about up to 100 volts approximately. But it's depending on which slice thickness it is, how big is this subject, and so on. And with the coil, It is especially so that the coil which you see there, this is used for data receiver or signal receiving. And the transmission is done by the big coil, which is under the cover of the magnet. So this is used for transmitter. Yeah? So you cover the whole body, more or less.
Let's continue with the questions for the candidate. Professor Železný.
So referring to one of the three facets of your contributions that you mentioned, which was the software development facet, I wonder if this was like pure software development, or it involved some sort of a research that would have a computer science character, let's say.
Yeah. Not purely research, not purely programming. So programming is a big part of that to make it all work, of course. But a large part is standard medical imaging or image processing. So all the things need to nicely work. There has to be some registration, segmentation. It's not that I would be developing new segmentation algorithms. That would be really a tremendous effort. But you need to put all these things together and make them work nicely. Plus,
so it was an application integration of, let's say, state-of-the-art image processing research methods, right?
Yeah, mostly. We did some extra development, because the data then needs to be quantified. You need to, properly, process the data, smooth them correctly, filter out noise. So there has been some methodological development, not necessarily,
And then if you were to say the single most important contribution of yours, would that be finding good application niches for the ASL?
Yeah, the most important, most useful to the community is to providing them with the software. Most useful scientifically is to find applications, make them work, and applying the method and finding new results of how perfusion works in the body.
Thank you.
Professor Tintiera.
Maybe I'd follow. Concerning of your contribution, I would say there is also one very important methodological research. This is this partial volume correction, and this is really very nice work. I like it very much. And so I also wanted to ask whether this technique which you develop and you just suggested is, let's say, generally useful, because the standard technique is to segment morphological data, T1 weighted morphological data, and then to use them to compensate for this partial volume effect. But you suggest to use low resolution data with T1 relaxation time calculation and so on, which is very nice and elegant method. But my question is, do you still think that this suggested method has also some restrictions, especially you must select some range of T1 relaxation time, it's based on some predictions and so on. Would you say that it is still generally useful for all possible resolution used in ISL, or you would say this is very good for if you have less of slices, low resolution, and maybe not so optimal, or maybe cannot be really optimal for higher resolution data with 3D acquisition with gradient spin echo sequence? Could you comment it? Which is because each method has some advantages, drawbacks.
I get your question. Sorry, that's a very specific question, so I'll answer to you. I'm not sure if all the others will be able to get it. So yeah, every method has its own drawbacks. So the main advantage of using to estimate the tissue components in the same resolution is that it is the exact same sequence. So you don't have any issues with the co-registration, deformations. It fits perfectly, and it brings a very nice image to it. And then, of course, it's the drawback between having a nicely working method that it's easily calibrated, but has different deformations, and it's not perfectly fit in the image, and the one that is perfectly fit in the image, but you need to focus on the exact relaxation times of the specific tissue and so on. I don't have the perfect answer because much more, it needs to be studied more to draw a definite conclusion. I think it works pretty nicely. So yeah, most of the challenges could be overcome by measuring the T1 time of the tissue and so on, but yeah.
Do we have any other questions? Maybe I would like to ask, the title of your lecture starts with the words processing and application, and the name of the method investigated follows. One may guess that the method is well established. How is established and what are the challenges for another development of this method compared to other known methods?
Yeah, so the measurement of ISL is pretty established, but there are new improvements to the sequence itself that are coming. So that's one thing. It can always be made better. We can measure more things to make it sure that it's better calibrated. And this also applies to the processing. While the standard is, yeah, pretty good, everybody uses the standard, there are more things you can do on the grounds of modeling. So you can improve the modeling of the signal measurement to have a better calibration and better quantification. So it's mostly about pharmacokinetic modeling of the blood, measuring all the necessary parameters to make sure that it's perfectly quantified.
Thank you. Yeah. So if there are no other questions, let me close the session. We will continue in a few minutes in room number 80 in the ground floor, and I kindly ask members of the habilitation committee and the reviewers to join. Thank you to the lecturer and the audience.